Hi everyone. Today we will discuss the drawing part of our question number two, where we are given a slide in the microscope and we are asked to make the plant diagram of the slide. Then they give us some part of the slide that we have to make under the high power diagram, what we call. And then the third part of the question is usually a comparison between the slide and a diagram that they give us in the paper. So let's start from the here. So the drawings actually, first of all, we must follow the instructions how to make the plant diagrams. Then we'll see what are the plant diagrams and how we should make them. Then diagrams and the comparison. So first of all, the instructions that we must follow before starting making the plant diagram. Our pencil should be sharp and thin. It should be as thin as zero point 0.7 mm, which is ideal for making the biology drawings. Thin less than 1 mm. There is no shading allowed. We should not shade any part of the diagram. The lines should not overlap. For example, if I'm making a line here, then another line is over here. So these lines are overlapping. We should take proper care that we make the lines smooth. There are no broken lines. Smooth line is like this one. And broken line, for example, is like this one. So if we have the broken lines over here. We must make the plant diagram showing the appropriate proportion of the each part or each tissue present in the diagram. We must compare the sizes of each tissue. If one tissue is 3 times thicker than the other, we must show it in the diagram. And it will depend upon the slide, how we observe it in the slide and make the plant diagram accordingly. Most of the times they don't tell us to make the plant diagram of the whole specimen. They just give us a part of it. For example, if the specimen is like this one, they tell us, okay, make the plant diagram of the half of it. Or they tell us to make the plant diagram of the 1/4 of this over here. So we must always make the plant diagram of only that part that is asked in the question. So let's move to the low power drawings, which is known as the plant diagrams. What are the things that we must remember before starting making a plant diagram? First of all, what we must do is this, there is always a space provided for making the diagram, plant diagram, and the marks is written over here, for example, 5 marks. We have to use this space properly, means we should use minimum 50% of the space. We should always use more than 50%. We cannot use less than 50%. Appropriate is if we use the most of the space that's given to us for making the diagram. Second is we must not write anything below this line where the marks are mentioned. And then the third is we must look into the paper and see what part of the slide we have to make the plant diagram of. Whenever we are making the plant diagram, we must always focus the slide and the objective lens should be at power 10. Eyepiece lens is already 10, so objective lens should be at power 10 when we are making a plant diagram. For example, I have taken the case of a root over here, ranunculus buttercup root, and it has the magnification of 35 that's given over here. For example, if you are asked to make the plant diagram of this part of the slide only, so they mention it that this part is to be made the plant diagram. That means we have to make the plant diagram of this part only. So I will make the outline first of all. Then we'll show the epidermis is here. So looking at the thickness of each tissue, I am making it accordingly. So this is the epidermis. Then I can see an air space is over here. There is some air space over here as well. And we can see a part of the endoderms here, and then there is the vascular tissue. This is what I can see under the slide of this specimen. And there is always also a part that they say use a ruled line to label this part. So we will use the scale to make a rule line. For example, if we are asked to show us where the epidermis is, so I will use a rule line, the scale to make this line. The line should actually start from where the tissue is. So the tissue is between these two lines. So I will write epidermis over here. Each tissue is basically shown by the two lines. So if we have epidermis from here to here, then we have cortex from here to here. Then we have endodermis from here to here. Then there is a vascular tissue inside it. Most of the times they tell us to show where the xylem is. So we'll show, okay, xylem is, if I can see it here, so I can see a part of it, xylem like this one. For example, this one is the xylem. This is the TS of a root and the part that they had mentioned as asked us in the question, and we have made a plant diagram of this part. No individual cell, nothing, we just show the tissues and their relative proportion. I've taken another example here, like the stem is over here. Let's say we have to make the plant diagram of the whole stem. So we can show that the stem has the epidermis over here. Then there are the vascular tissues, and the vascular tissues are of different size. I can see a big vascular tissue over here. There's a vascular tissue over here. There's a vascular tissue over here. The vascular tissue has the different tissues. We can clearly see it. For example, it's shown here like this. It has the tissue like this. The other one also has the differentiation like this one. And there seems to be a tissue from here to here as well. And it goes till here. This is the TS of the stem. If they ask us to show or label the xylem, okay, we'll make a ruled line and show this one is xylem, because in case of a stem, the xylem is toward the center of the stem and the phloem is towards the epidermis. Moving to the next part, this is the TS of a leaf, and for example, they tell us to make the plant diagram of this part only. So what shall I do? I'll make the two lines, and these two lines have the upper epidermis in between, so each tissue is to be shown by the two lines. Then there is the lower epidermis on this side. Try to make the lines as smooth as possible because I'm making it on the screen with the lip of the pen, so my hand is slightly shaky. Then we can see a tissue is over here, which is collenchyma densing, and then there is the vascular tissue. Vascular tissue mainly contains the xylem, and in case of the stem, or in case of the leaf, the xylem is towards the upper epidermis. So if I have to show the xylem, I'll make a ruled line and show this one is xylem. Now we'll move to the next part of the question, which is usually the high power diagrams. So whenever we have to make the high power diagrams, what we have to do is that we have to, it depends upon the question. If they tell us to make the four cells from, for example, a circle endodermis, or four cells, each cell touching at least two other cells, in that case we'll make the two rows. And whenever we are making the high power diagrams, we should shift the objective lens in the microscope to power 40 so that we can see the cells clearly. So for example, if we are asked to make the four cells from a ring of cells, so I will say this is one of the cells. The plant cell is always many-sided. This is another cell, this is another cell, and this is another cell. Each cell should be big enough. We say it should be minimum 4 to 50 mm having the diameter. And in case of the plant cell, each cell should always have a double line just to show that there is the cell surface membrane and there is the cell wall. And it should be as smooth as possible, as I am not able to make it so smooth on the screen. As I said, this is the writing on the screen is very tough, not so easy. So if we are asked to show or label the cell wall, so I'll make the line starting from the middle of the and write down this is the cell wall. So if this is a drawing of the four cells in the ring, now if we are asked to make the four cell drawing of the four cells and each cell touching at least two other cells. For example, I will show the two cells from the endoderms. It depends upon the instructions they give us in the question which cells we have to make. These are the two cells from the endoderms. And then there are the two cells inside it. We can see the xylem cells. So I have made these two cells like this one. As already discussed, we should always make the lines double in a plant cell to show the cell surface membrane and the cell wall. Then moving to the next part of the question usually is they give us a diagram in the paper and we have to compare that diagram with the slide we have in the microscope. Whenever you have a question about the comparison, we always make a table and this table has three columns in it. We should always write in the heading of the first column as the feature. Then the figure, for example, they give us in the paper, it is figure 2.2. And then we have in the microscope the slide J, slide K, whatever they call it. We never use the sentences to write the features or the differences between the two. So we always use the adjectives to mark the differences. For example, I say thickness of epidermis or epidermis. It was thicker in the figure 2.2 is thicker and here it is thinner than it. For example, if we see the shape of the figure 2.2 is not round, but in the slide J it was round. Number of xylem tissue. For example, here there were many xylem cells. Here there are only few. So we must write as many points differences as the marks are there. If the marks is there are 3 marks for the point, so we should write at least 3 differences. If 4, then we should write the 4 points. I hope this video will be very helpful to you for your exam. Thank you very much. Thanks for watching.